Chapter 1 Introduction to Cells. What does it mean to be living? People, petunias, and pond scum are all alive. Stones, sand, and summer breezes are not. What are the fundamental properties that characterize living things and distinguish them from non-living matter? The answer begins with a basic fact that is taken for granted by biologists now, but marked a revolution in thinking when first established 170 years ago. All living things are made of cells, small membrane-enclosed units filled with a concentrated aqueous solution of chemicals and endowed with the extraordinary ability to create copies of themselves by growing and dividing in two. The simplest forms of life are solitary cells. Higher organisms, including ourselves, are communities of cells, derived by growth and division from a single founder cell. Each animal, plant, or fungus is a vast colony of individual cells that perform specialized functions coordinated by intricate systems of communication. Cells, therefore, are the fundamental units of life. And it is to cell biology that we must look for an answer to the question of what life is and how it works. With a deeper understanding of the structure, function, behavior, and evolution of cells, we can begin to tackle the grand historical problems of life on Earth, its mysterious origins, its stunning diversity, and its invasion of every conceivable habitat. At the same time, cell biology can provide us with answers to the questions we have about ourselves. Where did we come from? How do we develop from a single fertilized egg cell? How is each of us different from every other person on Earth? Why do we get sick, grow old, and die? In this chapter, we begin by looking at the great variety of forms that cells can show, and we take a preliminary glimpse at the chemical machinery that all cells have in common. We then consider how cells are made visible under the microscope and what we see when we peer inside them. Finally, we discuss how we can exploit the similarities of living things to achieve a coherent understanding of all the forms of life on Earth, from the tiniest bacterium to the mightiest oak. Unity and diversity of cells. Cell biologists often speak of the cell without specifying any particular cell, but cells are not all alike. In fact, they can be wildly different. It is estimated that there are at least 10 million, perhaps 100 million distinct species of living things in the world. Before delving deeper into cell biology, we must take stock. What does a bacterium have in common with the cells of a butterfly? What do the cells of a rose have in common with those of a dolphin? And in what ways do they differ? Cells vary enormously in appearance and function. Let us begin with size. A bacterial cell, say a lactobacillus in a piece of cheese, is a few micrometers in length. That's about 25 times smaller than the width of a human hair. A frog's egg, which is also a single cell, has a diameter of about 1 millimeter. If we scaled them up to make the lactobacillus the size of a person, the frog's egg would be half a mile high. Cells vary no less widely in their shapes and functions. Consider the gallery of cells displayed in Figure 1.1. A typical nerve cell in your brain is enormously extended. It sends out its electrical signals along a fine protrusion that is 10,000 times longer than it is thick, and it receives signals from other cells through a mass of shorter processes that sprout from its body like the branches of a tree. A paramecium in a drop of pond water is shaped like a submarine and is covered with thousands of cilia, hair-like extensions whose sinuous beating sweeps the cell forward, rotating as it goes. A cell in the surface layer of a plant is a squat, immobile prism that surrounds itself in a rigid box of cellulose, with an outer waterproof coating of wax. A Delavibrio bacterium is a sausage-shaped torpedo driven forward by a rotating corkscrew-like flagellum that is attached to its stern, where it acts as a propeller. A neutrophil or a macrophage in the body of an animal crawls through tissues, constantly pouring itself into new shapes and engulfing debris, foreign microorganisms, and dead or dying cells. Some cells are clad only in a flimsy membrane. Others augment this delicate cover by cloaking themselves in an outer layer of slime, building themselves rigid cell walls, or surrounding themselves with a hard, mineralized material, such as that found in bone. Cells are also enormously diverse in their chemical requirements and activities. Some require oxygen to live. For others, it is deadly. Some consume little more than air, sunlight, and water as their raw materials. Others need a complex mixture of molecules produced by other cells. Some appear to be specialized factories for the production of particular substances, such as hormones, starch, fat, latex, or pigments. Some are engines, like muscle, burning fuel to do mechanical work. Others are electricity generators, like the modified muscle cells in the electric eel. Some modifications specialize a cell so much that they spoil its chances of leaving any descendants. Such specialization would be senseless for a cell that lived a solitary life. In a multicellular organism, however, there is a division of labor among cells, allowing some cells to become specialized to an extreme degree for particular tasks and leaving them dependent on their fellow cells for many basic requirements. Even the most basic need of all, that of passing on the genetic instructions to the next generation, is delegated to specialists, the egg and the sperm. Living cells all have a basic chemistry. Despite the extraordinary diversity of plants and animals, people have recognized from time immemorial that these organisms have something in common, something that entitles them all to be called living things. With the invention of the microscope, it became clear that plants and animals are assemblies of cells, that cells can also exist as independent organisms, and that individual cells are living in the sense that they can grow, reproduce, convert energy from one form into another, control their internal workings, respond to their environment, and so on. But while it seemed easy enough to recognize life, it was remarkably difficult to say in what sense all living things were alike. Textbooks had to settle for defining life in abstract general terms related to growth and reproduction. The discoveries of biochemistry and molecular biology have made this problem disappear in a most spectacular way. Although they are infinitely varied when viewed from the outside, all living things are fundamentally similar inside. We now know that cells resemble one another to an astonishing degree in the details of their chemistry, sharing the same machinery for the most basic functions. All cells are composed of the same sorts of molecules that participate in the same types of chemical reactions, discussed in Chapter 2. In all living things, genetic instructions, genes, are stored in DNA molecules, written in the same chemical code, constructed out of the same chemical building blocks, interpreted by essentially the same chemical machinery, and duplicated in the same way to allow the organism to reproduce. Thus, in every cell, the long DNA polymer chains are made from the same set of 4 monomers, called nucleotides, strung together in different sequences like the letters of an alphabet to convey different information. In every cell, the instructions in the DNA are read out or transcribed into a chemically related set of polymers called RNA. RNA molecules have a variety of functions, but the major class serve as messenger RNA. The messages carried by these molecules are in turn translated into yet another type of polymer called a protein. Protein molecules dominate the behavior of the cell, serving as structural supports, chemical catalysts, molecular motors, and so on. Proteins are built from amino acids, and every living thing uses the same set of 20 amino acids to make its proteins. But the amino acids are linked in different sequences, giving each type of protein molecule a different three-dimensional shape, or conformation, just as different sequences of letters spell different words. In this way, the same basic biochemical machinery has served to generate the whole gamut of living things. Figure 1.3. A more detailed discussion of the structure and function of proteins, RNA, and DNA is presented in chapters 4 through 8. If cells are the fundamental unit of living matter, then nothing less than the cell can truly be called living. Viruses, for example, are compact packages of genetic information in the form of DNA or RNA, usually encased in protein, but they have no ability to reproduce themselves by their own efforts. Instead, they get themselves copied by parasitizing the reproductive machinery of the cells that they invade. Thus, viruses are chemical zombies. They are inert and inactive outside their host cells, but exert a malign control once they gain entry. All present-day cells have apparently evolved from the same ancestor. A cell reproduces by duplicating its DNA and then dividing in two, passing a copy of the genetic instructions encoded in its DNA to each of its daughter cells. That is why daughter cells resemble the parent cell. However, the copying is not always perfect, and the instructions are occasionally corrupted by mutations that change the DNA. That is why daughter cells do not always match the parent cell exactly. Mutations can create offspring that are changed for the worse, in that they are less able to survive and reproduce. Changed for the better, in that they are better able to survive and reproduce, or changed neutrally, in that they are genetically different but equally viable. The struggle for survival eliminates the first, favors the second, and tolerates the third. The genes of the next generation will be the genes of the survivors. Intermittently, the pattern of descent may be complicated by sexual reproduction, in which two cells of the same species fuse, pooling their DNA. The genetic cards are then shuffled, redealt, and distributed in new combinations to the next generation, to be tested again for their survival value. These simple principles of genetic change and selection, applied repeatedly over billions of cell generations, are the basis of evolution, the process by which living species become gradually modified and adapted to their environment in more and more sophisticated ways. Evolution offers a startling but compelling explanation of why present-day cells are so similar in their fundamentals. They have all inherited their genetic instructions from the same common ancestor. It is estimated that this ancestral cell existed between 3.5 billion and 3.8 billion years ago, and we must suppose that it contained a prototype of the universal machinery of all life on Earth today. Through mutation, its descendants have gradually diverged to fill every habitat on Earth with living things, exploiting the potential of the machinery in an endless variety of ways. Genes provide the instructions for cellular form, function, and complex behavior. A cell's genome, that is, the entire library of genetic information in its DNA, provides a genetic program that instructs the cell how to function, and, for plant and animal cells, how to grow in an organism with hundreds of different cell types. Within an individual plant or animal, these cells can be extraordinarily varied, as we discuss in Chapter 20. Fat cells, skin cells, bone cells, and nerve cells seem as dissimilar as any cells could be, yet all these differentiated cell types are generated during embryonic development from a single fertilized egg cell, and all contain identical copies of the DNA of the species. Their varied characters stem from the way that individual cells use their genetic instructions. Different cells express different genes. That is, they use their genes to produce some proteins and not others, depending on the cues that they and their ancestor cells have received from their surroundings. The DNA, therefore, is not just a shopping list specifying the molecules that every cell must have, and a cell is not just an assembly of all the items on the list. Each cell is capable of carrying out a variety of biological tasks, depending on its environment and its history, using the information encoded in its DNA to guide its activities. Later in this book, we shall see in detail how DNA defines both the parts lists of the cell and the rules that decide when and where these parts are to be made. Cells under the microscope. Today, we have the technology to decipher the underlying principles that govern the structure and activity of the cell. But cell biology started without these tools. The earliest cell biologists began by simply looking at tissues and cells, then breaking them open or slicing them up and attempting to view their contents. What they saw was to them profoundly baffling: a collection of tiny and scarcely visible objects whose relationship to the properties of living matter seemed an impenetrable mystery. Nevertheless, this type of visual investigation was the first step toward understanding, and it remains essential in the study of cell biology. Cells in general are very small, too small to be seen with the naked eye. They were not made visible until the 17th century, when the microscope was invented. For hundreds of years afterward, all that was known about cells was discovered using this instrument. Light microscopes, which used visible light to illuminate specimens, are still vital pieces of equipment in the cell biology laboratory. Although these instruments now incorporate many sophisticated improvements, the properties of light itself set a limit to the fineness of detail they can reveal. Electron microscopes, invented in the 1930s, go beyond this limit by using beams of electron instead of beams of light as the source of illumination, greatly extending our ability to see the fine details of cells and even making some of the larger molecules visible individually. A survey of the principal types of microscopy used to examine cells is given in panel 1.1 in pages 8 and 9. The invention of the light microscope led to the discovery of cells. The development of the light microscope depended on advances in the production of glass lenses. By the 17th century, lenses were refined to the point that they could be used to make simple microscopes. Using such an instrument, Robert Hooke examined a piece of cork and in 1665 reported to the Royal Society of London that the cork was composed of a mass of minute chambers, which he called cells. The name cell stuck, even though the structures Hooke described were only the cell walls that remained after the living plant cells inside them had died. Later, Hooke and his Dutch contemporary Antony van Leeuwenhoek were able to observe living cells revealing a previously unseen world teeming with motile microscopic organisms. For almost 200 years, the light microscope remained an exotic instrument, available only to a few wealthy individuals. It was not until the 19th century that it began to be widely used to look at cells. The emergence of cell biology as a distinct science was a gradual process to which many individuals contributed, but its official birth is generally said to have been signaled by two publications, one by the botanist Matthias Schleiden in 1838 and the other by the zoologist Theodor Schwann in 1839. In these papers, Schleiden and Schwann documented the results of a systematic investigation of plant and animal tissues and the light microscope, with the light microscope, showing that cells were the universal building blocks of all living tissues. Their work and that of other 19th century microscopists slowly led to the realization that all living cells are formed by the division of existing cells, a principle sometimes referred to as the cell theory, figure 1.4. The implication that living organisms do not arise spontaneously, but can be generated only from existing organisms, was hotly contested, but it was finally confirmed by experiments performed in the 1860s by Louis Pasteur. The principle that cells are generated only from pre-existing cells and inherit their characteristics from them underlies all of biology and gives the subject a unique flavor. In biology, questions about the present are inescapably linked to questions about the past. To understand why present-day cells and organisms behave as they do, we need to understand their history, all the way back to the misty origins of the first cell on Earth. Darwin's theory of evolution, published in 1859, provided the key insight that makes this history comprehensible, by showing how random variation and natural selection can drive the production of organisms with novel features, adapted to new ways of life. This theory of evolution explains how diversity has arisen among organisms that share a common ancestry. When combined with the cell theory, it leads us to a view of all life, from its beginnings to the present day, as one vast family tree of individual cells. Although this book is primarily about how cells work today, we shall encounter the theme of evolution again and again. Cells, organelles, and even molecules can be seen under the microscope. If you cut a very thin slice of a suitable plant or animal tissue and place it under a light microscope, you will see that the tissue is divided into thousands of small cells. These may be either closely packed or separated from one another by an extracellular matrix, a dense material often made of protein fibers embedded in a polysaccharide gel, figure 1.5. Each cell is typically about 5 to 20 micrometers in diameter, figure 1.6. If you have taken care to keep your specimen under the right conditions, you will see that the cells show signs of life. Particles move around inside them,